Okay, welcome everyone. My name is Brenton Clark and I'm the president of the local branch here. We have with us a dignitary from King's College, University of London. Cathryn is the winner of the Frank Hanford Miller Fellowship for 2019, and as part of the bequest which Frank left, Frank was into medals. He liked to go in marathons and he liked to talk about the medals they had at the RSS. So in a tradition that was set by Frank for all his medals, Cathryn gets a medal. Cathryn Lewis, Frank Hanford Miller Fellow on the other side, 2019. Cathryn's going to tell us a bit about herself.
Okay. Great. So. Thank you, Brenton. Thank you, everyone. It's been wonderful to be in Perth for the last 10 days, and I've got a few days left this week before I fly out to Sydney on Saturday. And it's been just delightful. Thank you to everyone for their hospitality and the interesting discussions we've had. What I'm going to talk about is my research, which is very much on the applied end of statistics, but working on genetics, which I know is of interest to some people in the audience, but I'm going to run through that. But before I did that, I thought I'd introduce myself. So I'm currently a professor at King's College London. My academic background is in maths and statistics, so I did a bachelor's at Oxford, and then a master's and PhD at the University of Sheffield, which is a very strong statistical department there. And then I did a postdoctoral role at the University of Utah in Salt Lake City. And I was working with the group there that found the BRCA1 gene that increases risk of breast cancer. And that was a hugely exciting time in genetics, because for the first time, we found genetic mutations that had a direct role in assessing risk. But the US wasn't home, and so I came back to the UK after about 5 years to a faculty role at King's College London, and I've been there for the last 20 years, although 10 of those in the middle I worked part-time whilst having children. And I've put that picture up. So this is my office building here. I have an office on the 8th floor there, with a wonderful view over London, and as one of the audiences just pointed out, if you go on my Twitter page here, the skyline from my office is there. So, and I partly mention that to say that we always have a lot of statistical jobs going, and particularly in statistical genetics, and we really aren't training enough numerate young people to fill all the opportunities that are available, and so if you'd like to work with this view too, please get in touch. And so, as Brenton said, here I was very honoured to be appointed the Frank Hanford Miller Fellow for this year, and I read in detail Brenton's article summarising his interview with Frank back in 2002. And I was struck by this quote at the time, that he was a rather eccentric Englishman who adopted Australia as his home after he retired from the UK. And I sort of read that in London with, oh, interesting. But now I've come here, I can see exactly why he did that. And if you've got tutorial posts going in 10 years' time, I might well be interested. So, what I'm going to cover here, I'm going to give a bit of an introduction to genetics and polygenic risk scores so that we're all on the same page there. I'm going to talk about the statistical analysis methods and then show how these are valuable in applications, some of the challenges, and come back to Frank Hanford Miller at the end. And we're a fairly small group, so I'm very happy to take short questions, particularly points of information as we go through. So I've got just a few slides introducing us to DNA. So this is the discovery of the structure of DNA. by Watson and Crick in 1953 in Cambridge with their model there. And so they essentially found this double helix here and the way that the four bases were structured there. But when you're at King's College London, the person that is missing from that picture is Rosalind Franklin here, who was her crucial picture of DNA, the X-ray picture that enabled them to discover that structure. And I quite like this picture here. It's a Quentin Blake one from Addenbrooke's Hospital in Cambridge. If you're ever there, there's a whole frieze of similar pictures along the hospital corridor, which are excellent. But I quite like this idea that she's the one doing the really detailed hard work, and they're the ones in the park drinking beer and discussing it. And I don't think science has changed terribly much in some ways in the last 50 years. So why is a statistician interested in DNA? Well, our whole DNA is 3 billion bases of those ACGT bases on this ladder. And that's a huge amount of data, 3 billion base pairs. If you store it, it takes 3 gigabytes of storage space. It'll take about 50 years to type out 1 letter at a time. So that's a huge amount of data there. Now, most of those 3 billion base pairs are exactly the same between all of us. So if I have an A in one spot, then Graham will too, and Rick and all people through the audience. But that's true for 99.9% of our DNA, but that 0.1%, we carry different base pairs, and those are base pairs that are inherited through the family, and those are differences that make us who we are. So it's those differences that control our hair colour, our height, the diseases that we get, all sorts of physical characteristics and our mental health. And of course 0.1% doesn't sound very much there, but 0.1% of 3 billion is quite a large number. And what we've been doing, what my research has been involved in, is trying to identify which of those DNA differences contribute to disease risk and in what way. So the model that we have for most genetic disorders looks like this. And I'm putting to one side the rare Mendelian single gene disorders that we learn about at school, like Huntington's disease and cystic fibrosis, where we're just talking about a single gene and a variant there increases risk. Most of the diseases that are a real public health problem fit into this sort of model where we have many different genes, genetic variants contributing. There'll also be environmental risk factors contributing and possible interactions within and between those. And the idea is that it's not until we know about all of this that we could get a good estimate of whether someone is likely to become affected with a disorder or not. And obviously the relative balance between environmental risk factors and genes will depend on the disorder. So lung cancer, for example, will have a huge environmental risk factor of smoking, but genetics is also relevant. And most disorders that have this major impact, this economic, social, or health burden for sufferers and their families, have this sort of model. And we've known for quite a few decades about many of the environmental risk factors, but it's only in the last decade that we've started to identify and chip away at what the genetic component to those disorders are. So I'm going to run through in a little bit of detail exactly how that works to get a feel for it and then step back a bit. So I talked about these DNA places where we vary. So this is a sort of section of a chromosome here. And you can see the first few bases are exactly the same in both those chromosomes, T, G, G, A, C. But then the next one varies. There's a T here and a C over here. And those will be inherited by the next generation. And we say that alleles, forms, if you like, T and C are present in the population. But of course, each of us carry 2 copies of those, one that's inherited maternally and one that's inherited paternally. So that when we're analysing data at a people level, we need to consider two of those. So someone might carry 2 copies of the T allele, 1 each of a T or C, or 2 copies of the C allele. So that's the basic data that we're analysing. It comes into 3 categories, and you can calculate the probability of someone carrying each of those. And then what we're really interested in is to say, well, is there evidence of any of these places where there are different alleles, so these dots mean there's no change there. We test the hypothesis that at this site here, where there's a T and a C allele, that cases with depression, which is what I'm interested in, but it works for any disorder, whether they are more likely to carry a C allele than a T allele at that locus. So it's a very simple hypothesis of whether cases with depression have a different genetic distribution of these genotypes here than controls who don't have depression. So what that data looks like is that you would genotype a set of cases who have the disorder, a set of controls who don't, classify them into these 3 groups, and then you can see just eyeballing that there's more of the red C alleles in the cases than in the controls, suggesting that that C allele, whether it's in a pair or as a heterozygote combined with a T allele, carrying a C allele increases your risk of disease. You're more likely to develop depression. Now, that's a simple, just eyeballing it. What we actually do now, that genetic technology has so far advanced, is that we don't look at one site at a time, but we do this across our whole genome. And we have standard DNA chips that will look at 500,000 of these sites at one time. So we collect our DNA from our cases and controls, so we collect a blood sample or a saliva sample, we extract the DNA, and then we genotype it on these chips. And so what we end up with is this data matrix at the bottom here. So each column is an individual's genotype, so there will be 500,000 rows in that matrix, and then, 500,000 rows, and then columns for each individual. And then what we do is we take each row here and test in turn whether the distribution of genotypes in the cases differs from the distribution in controls. And you might think in this age of data science and machine learning that we could just throw this whole matrix at a machine learning package, and it would come back and tell us what the answer was and which sites on DNA differ between cases and controls. But in practice, we found that the most powerful way to analyse this is not as a complete matrix in that way, but it is to perform 500,000 hypothesis tests, analysing each row at a time, testing the hypothesis that the genotypes differ between cases and controls. And we usually do that in a logistic regression framework, so we're trying to predict case control status on the SNP genotypes, which we just code as 0, 1 or 2 for the number of alleles that people carry. And then we have to do a bit of adjustment for ancestry to account for the fact that our cases and controls might differ, not just because they're affected with a disorder or not, but because they come from slightly different ancestral groups. And what that gives us is it gives us an odds ratio for the SNP, the increased risk for each allele carried. And it does that for at least the 500,000 SNPs that we've tested. And because of that, we have a multiple testing issue and to declare significance, we want to achieve a p-value of 5 × 10⁻⁸. So that's controlling for the 500,000 tests that we've done. Okay? And obviously to get a p-value that's that significant, we need large sample sizes to be able to attain that. So I've been working on the genetics of depression for the last 12 years or so. And if you're familiar with genetic studies from other disorders like type 2 diabetes or breast cancer or cardiovascular disorder, autoimmune disorders, they started finding genes back in this period, as soon as we started doing these genome-wide studies. For depression, it was much more difficult, and I'm going to talk a bit about that. And we didn't find our first hits until 2015, but since then we have steadily moved upwards. So we now have 102 hits genome-wide, 2 studies that have been published in the last 2 years. And what these graphs show, they're what's called Manhattan plots. Across the x-axis are the chromosomes, chromosome 1 to 22, and each point is one genetic variant that's been tested, so one hypothesis that's been tested, and the y-axis here is minus log p-value, so high values are significance. Most SNPs, most results are right down by the axis here that show no evidence of association. In the plot on the left, the green diamonds are SNPs that are associated, but you'll see most of those have a column of other variants underneath that are associated, and that's just to do with correlation across regions of the genome. So the diamonds are the ones that are important, and the others are just correlated supporting variants, but not independent signal for disease. So that's great. We, over the last few years, have made enormous progress in identifying the genes for depression. We now have 102 different variants that give us some biological insight to what might be happening in the body, in the brain, to put someone at increased risk of depression. But each of these variants have a very small effect on risk. So individually, they tell you nothing about whether someone is likely to develop a disorder or not. But we have discovered that by pulling them together, by looking genome-wide at what people carry, that can give us some information on risk. And that's the Polygenic Risk Score that was in the title of my seminar. So what we do there is we take this discovery study on the left-hand side, where we've identified the variants that are associated, and then we take that information and we put it into a separate target study that has to be completely independent for reasons I'm sure I don't need to explain to you. And then within that study, we construct a polygenic risk score for every individual that gives you their liability, their genetic liability to disease. And we do that just with a weighted sum here, which I'm going to come back to on the next slide. But the really nice thing about this is that it takes this massive matrix that we started with of 500,000 SNPs across everyone in our study, and it pulls that down to one number for each individual that captures their genetic information. So what we do is, as I said, it's a weighted sum. So for each individual, they have a polygenic risk score for the disease, and we sum across the SNPs that we choose. So X is the 0, 1 or 2, the genotypes that someone carries, and the beta is the loading for that disorder, for that SNP. So we usually use the log odds ratio as an effect size, which just means that if a locus has higher evidence of effect, it contributes more to this weighted score. And there's a few different ways that we can do this. I should say, perhaps coming back to this Manhattan plot, this red line here is our genome-wide association threshold for significance of 5 times 10 to the minus 8. And so you could say, great, I'll just take those green diamonds above that line, and sum just those SNPs that I know are statistically significant. But we've discovered that what's often called the grey zone, just below that line, also carries signal, and that the next time we do a bigger study, those SNPs will have moved up over the line. And so we're just lacking power to show many of the SNPs in this region contribute. And so what we can do is move that red line further and further down until we're including all the SNPs. And this is what one of the methods that we use for calculating polygenic risk scores does. It's, the software is called Precise. I wrote this with a PhD student and my colleague Paul O'Reilly about 4 years ago. And it's just very simple. It removes correlated SNPs to make SNPs independent because we're summing over them. And then it uses a whole range of different p-value thresholds. It starts with just those significant ones and then moves it down until it's including all independent SNPs, which is usually about 100,000 across the genome. And then it calculates scores at each of those about 10 different p-value thresholds. And that's very simple. It's very intuitive. You can see exactly what you're doing. And it would be easy to implement clinically because that's really what we would like to do ultimately, is to be able to use this genetic information to identify who is at high risk for different disorders before they get it, so that they could have screening, that they could, and I'll talk a bit more about that in a minute. So that's the most simple method. It's not the statistically most powerful method, and there are various different pieces of software out there now. LDPRED is probably the most widely used one, which instead of removing the correlated variants, it models them and it retains all the SNPs in the model, which seems to perform better than Precise does. But it's quite a black box method, and so, you know, for research studies it works great. I think as we try to move things towards the clinic, the criticism that you often hear is, well, do we really need information on 8 million genetic variants to identify someone's genetic risk? And then stepping even further in that direction, because this is really just a machine learning problem. How can we pool together information on these variants across the genome and do some sort of joint estimation of effect size and variance? And there's a lot of work going on in this area, and I know that's what Novia is working on in her PhD with Nicola. And it's a very good statistical approach to the problem. I think it's not straightforward, particularly, you know, our variables are, you know, 0, 1 or 2. And we have a lot of collinearity, a lot of correlation, and even the classic machine learning methods, which are supposed to get around this, in practice we find don't work very well. And certainly when we've tried to apply machine learning methods to this or to the discovery, our experience has been that there's too much noise and not enough signal, I think, to be able to easily cut through this. So I think we need some more research in that area as exactly how to use these methods most appropriately, and probably feature selection as an independent step to cut down the number of variables are quite useful. So the methods I'm going to talk about are the Precise software in the left-hand column, partly because it's most intuitive, it's also most widely used. This software has now got a second version from Sam Choi and Paul O'Reilly. If you're calculating polygenic risk scores in this method, I highly recommend it. It's fast, it's flexible, it's scalable, so this is across numbers of samples here in time, and you can see that LDPRED suffers substantially as the sample sizes get large, whereas Precise remains quite feasible to carry out. And geneticists, we tend to like practical things. We like things that work, that we can get things done and move them into the next, move them into using them. So, coming back to polygenic risk scores, it probably won't have escaped your notice that because we're doing this weighted sum and the n, the number of SNPs that we're adding across, is very large, each one having a small effect, then what you will end up with in the population is a normal distribution of scores. And what that means is that most people in the population will have a score that's very close to the average. So actually knowing their genetics won't tell you anything about their risk of disease. It's still the sort of the prevalence in the population. Is the genetics doesn't add much there. Of course, people down this end of the distribution will have decreased risk because they carry fewer risk alleles than you might expect. And the one that we're usually interested in are at the other end, those that have an increased risk because they carry many of those risk alleles. And you can imagine that one of the things we might want to do is to identify those in the top 1% or top 5% of the distribution that we might want to offer early screening to, or other sort of therapeutic interventions to help them avoid getting the disorder. So we have these scores. We can calculate them in any one that we have genotype data for. And these are the sorts of things that we've been doing on the research side with them at the moment. So what we might want to do is to say, well, we've got these scores in our research studies. Do they actually predict? How well do they predict cases and controls? So for example, as I have 2 boxes on a previous slide, our depression polygenic risk scores, does it predict case control status in a separate study? And then you might also want to look across disorders. So, for example, our depression polygenic risk scores, part of that is just about depression, but part of the variants are also relevant more broadly to other mental health disorders. And so those polygenic risk scores also predict anxiety, for example, and bipolar disorder. So how big is the signal there? And then the third thing that we might want to do is to dissect heterogeneity within the trait. So do these scores predict who's going to have an early onset case, who's going to have a severe case, who's going to be resistant to treatment? Can we start to subdivide our sort of single box of cases using genetics? And that ties up very nicely with something that Frank Hansford-Miller said. He said we've become too technical. We should ask, how is this piece of research going to help humanity? And my real goal in the next 10 years of my working life is to take these scores from a research setting to where they could be useful in clinical care. You can invite me back in 10 years' time and I'll tell you how successful I've been or not. So the way that we do these sorts of studies is another logistic regression, because again we're trying to predict case control status here, where P here is the probability that someone is a case within that study. And this time we're testing, we don't have a SNP genotype, we have the continuous polygenic risk score there. We're still correcting for ancestry and genotyping things that might explain some of the difference between cases and controls. And we're testing this null hypothesis that beta is 0. And then we're often also wanting to estimate the odds ratio for disease, so how much increased risk of disease per one standard deviation of the polygenic risk score there. And you can see how that works. So in the population, our scores have a normal distribution. If we look at cases, what we would really like to see is that our case distribution was right up here, but sadly we're not that far yet, and what you see is a very subtle shift in the mean polygenic risk score between cases and controls. But the way that these scores get power is if you look at the tails of the distribution, you can see that there's proportionately quite a difference in the tails, and that's where this is really useful. And that makes sense actually, because these people in the middle here, I've said their genetics doesn't tell you very much anyway. They're the ones that are very close to the population average, and so it's the tails that we're really interested in. So putting that together, we run our logistic regression. We get our and we get parameters that come out of that. How do we measure what the effect size is? Whether the cases and controls really differ. So there's a lot of different outcome measures that you can use. So you can use, as I've said, the odds ratio of disease risk per one standard deviation of the polygenic risk score. You can look at the just the tails of the distribution and see how they differ in disease risk. You can look at the model level, it's logistic regression, so you could look at Nagel-Kirkey's R-squared, how much of the case control variation does that explain, or then you need to convert that to the liability scale, I'll come back to that. Or we can use the AUC, so that's a measure of how likely we are to distinguish a case and a control correctly. So I'm going to sort of work my way up the list. So what the AUC does is it measures how separate the two distributions for cases and controls are here. And it plots this graph down here of sensitivity, y-axis against 1 minus specificity on the x-axis. And it does this by moving this vertical cutoff through the distribution. So it starts here and moves its way downwards. And you get a graph that is anchored at each end of this diagonal line, so it starts at 0, 0, ends up at 1, 1. And a really powerful discriminatory test, where the distributions are well separate, would go from 0, jump straight up to 1, and across the top. Do that. Whereas a test with no predictive value at all, just random chance, would follow this diagonal line through the middle here. And because of the way that works, the measure that people have come up with for summarising the fit, the predictive ability rather, is to look at the area under this dotted line for the test, which varies from a half, if it were the line of no fit here, random chance, to 1 if it has a perfect fit. Now AUC, because it goes between a half and 1, people find it quite difficult to interpret. People think of it as a correlation. It's not a correlation, of course, because the minimum value is 0.5 rather than 0. But the easy way to interpret that is to say that what the AUC actually tells you, if you had a value like 0.7, it would mean that if you had one person who was a case and one person that was a control, there was a chance of 0.7 that the case would have a higher score, polygenic risk score, than the control. So it's a very simple summary measure there. But of course, to have a test that is clinically valid, clinically useful, 0.7 is not usually good enough to distinguish those two. You want something that is much closer to 0.9 or 0.95. Okay? And most of the values that we're getting are 0.5, 0.6, 0.7. So polygenic risk scores have quite a long way to go. The other way that you can think about this is how much of the variation is explained. So in Nagel-Kirkey's R-squared, you have to do a bit of tweaking here, because what comes out of the model is what you've actually analysed, which is your set of cases and controls. But because we've done ascertainment usually, these are usually not population studies, we've over-ascertained our cases compared to their prevalence in the population. And Nagel-Kirkey's R-squared tells you about what you've actually got in your study, whereas what you really want is how much at a population level does this test explain, and you have to do a bit of conversion to get from this observed scale onto the liability scale, where your cases are just represented by their population prevalence. And there's a very good paper from Naomi Ray's group which goes through how to do that and what these measures actually mean. So, but in practice this is really quite simple, because it's just about normal distributions. So our normal distribution in the population, I've standardised it without loss of generality, so mean 0 and standard deviation of 1. Our controls will have a distribution that's slightly to the left, because they're not affected with disease, and you can figure out where the mean value is there. Our cases will be up here, and that's really just controlled by two things. Cohen's D of how far the case mean is shifted upwards, and the prevalence of the disorder. And from those two measures, you can figure out exactly what you need about the fit of polygenic risk scores. And you can also then do things like figure out, well, what's the rate of disease in the lowest, the population that have the lowest 10% of, so that should be 10% and then the highest 10% here. And you can just figure that out exactly from those two parameter values. And so you can flip backwards and forwards between Cohen's d disease prevalence with a bit of assumption about equal standard deviations in cases and controls, and then you can find out the AUC, the odds, any odds ratios that you want, and an expected R squared on liability. So it's fairly simple statistical manipulation. And so, for example, with a Cohen's d of 0.2, you get an A, which is quite large by some of these studies I've done. You get an AUC of 0.55, so that's only slightly over random chance. And then if you compare the deciles at the top and bottom 10%, there's an odds ratio of just over 2 that someone will be affected given they have a polygenic risk score at the top compared to the bottom decile there. So there's a fairly simple. But these studies, of course, are based on research studies of people that have been specifically selected to figure out what's going on in the disease. If we really want to move this towards clinical care, then we have to be able to interpret that for an individual. We have to be able to say to an individual, your score is this is what it means in terms of your disease risk. And I'm going to walk through some examples showing you where polygenic risk scores might ultimately become useful in the clinical pathway. So because they're inherited genetics, you can calculate them from birth. Now, that's not a good idea, but I just want to make the point that our genetics is fixed. You might want to do it when determining who needs to be screening for a disease, when someone shows symptoms, at a point of diagnosis where there may be many differential diagnoses possible, in determining treatment, or trying to predict course of disease. So starting at the beginning, predicting birth, this is how well that works or doesn't work for depression. So in our depression study back in 2018, we calculated polygenic risk scores, and they explain just 2% of the variance in disease risk. So although we're hugely excited to finally be identifying genes that are associated with depression, it's just the tip of the iceberg. Even when you put them together, they don't explain very much. And if you do the decile comparison, those with the highest decile have a 2.5-fold increased risk compared to those in the lowest decile, which may not sound very much, but because remember, depression is quite a common disorder, has a prevalence of about 15%. So converting those to absolute risk might still be a reasonable risk. And then it does sensible things that you see higher scores in cases that are recurrent, are more severe, or have earlier onset. So overall, a very modest capture. I think the AUC is about 0.55 here, but clearly capturing some signal, not just noise. Other disorders have done better. So in coronary artery disease, for example, they've got to the point where they're starting to say, these are useful, we should be using them clinically. Cathryn Lewis said, you know your cholesterol, your blood pressure, you should also know your heart gene score. And how long will it be before that's part of standard healthcare? Well, you can get that already if you've got your genotypes. This is an app that I had on my phone. It uploaded my genotypes, and this is my risk for coronary artery disease. On this bar here, I'm at the 65th percentile, and even I saw that and went, oh, but actually it's just over the middle for that normal distribution. So, but it was quite telling that even I, who knew what this score meant, it was very easy to misinterpret this when I first saw it. So that's this is an app that I can get on my phone, and I think in this case there is some information there. I was rather more surprised to check out Gene Plaza, which pulls together different genetic testing apps, and they offered to tell you the genetics of what your risk was for depression, this depression that accounts for 2% of the risk. This is the graph that they show. I haven't uploaded my genotypes to this website, This is one of those graphs that would be quite good in a stats paper to try and get people to show exactly how many ways this is wrong. The numbers up the side, that's not what a normal distribution does. We cannot figure out where they get the numbers at the bottom from at all. You can't do that. And then this 4% at the top is a population prevalence, point prevalence. So I wasn't giving them my data. But you notice that this is your risk for depression. You notice they put you at a low risk of depression. They also do this for IQ, and you can imagine that the example they give, you're right up here with your pseudo IQ score. So, but the reason that this is just nonsense, apart from the fact that they clearly don't understand statistics, is that there's no information there. So our liability to disease, we can partition into what we know is the polygenic risk score, what we don't know, the unknown. The polygenic risk score is a variance of 2%. That leaves 98% that is unknown. So that means that even someone whose score is quite high on the normal distribution and known quite accurately, you're combining that with 98% of random noise, which will completely swamp that signal. So just not really a very good idea to do. If you do want to look at your polygenic risk scores, I can highly recommend this site from Lasse Folkesson, who's a statistical geneticist in Copenhagen. And he got interested in this. So this is Lasse, this is his wife, who is Chinese, and this is their daughter, who has blonde hair. And he said, how does that work? And he started to look at genetics and ended up building a polygenic risk score estimator. You just upload your data to his website. He figures out what he needs from it. He throws away what he doesn't need, so there's very few security issues. And then he will calculate polygenic risk scores. And he shows this, just as I've done on a normal distribution graph, and shows you where you lie. And this site's been quite popular. He's got over 25,000 unique users who've done over 3 million requests. And one of the things he does is he orders his traits alphabetically. Some sites order them by risk, so they'll give you your highest risk first, but he says you can't do that. There's not enough information to justify that. But that does give you good information on what it is that people want to find out. So these are the scores that the users seek. Now remember, these are alphabetical, so people click on the top few on the list just to see what this is about, and that's probably the ones above that red line there that are just the alphabetical ones. But below that, you can start to see the disorders that people are choosing to look at. Obesity, type 2 diabetes, stroke, so cardiometabolic, quite common in the population, not too surprising. What really did surprise me was the number of mental health disorders on that list. So schizophrenia, bipolar, depression, they're not very strong, but those are the ones that people are really interested in finding out about. And also, and Alzheimer's is quite high on that list too. So, people really are interested in this. They really want to know what genetics can tell them. And one of the things that I think it might tell us most quickly is how we should optimize screening protocols for people. So this is breast cancer. I moved on from depression. And the polygenic risk score for depression for breast cancer is much more informative. The bottom 20% have a 5% risk compared to 16% in the top 20%. The top 1% have a threefold increased risk, which again, given breast cancer, certainly in the UK is about 10% prevalence, a threefold increased risk on that, really makes it quite high. And of course, we have a screening program for breast cancer, mammograms, and it's very simple. It's run very simply. We stratify by age, and we stratify by sex. And the question comes whether we should also be stratifying by genetics. Because it's a very popular screening program, certainly in the UK, and I think here also. But it's problematic that there are a lot of false positives, a lot of unnecessary biopsies, a lot of follow-up tests, a lot of increased anxiety. If we could focus the screening on women who were at higher risk for breast cancer, it would make it a much more effective test, and it would make it probably easier to show that it really does save lives, which is marginal at the moment. And this graph shows you that that would work. So each line is a sort of threshold on the polygenic risk scores. So the middle bit here, 40 to 60%, that's the middle 20% of the distribution, that's average. X-axis is years, and then Y-axis is 10-year absolute risk, which is what the screening program is worked out at. And it's designed that when you hit that red line, which average counts just before the age of 50 here, that's when screening kicks in. And on average, that works, if you look at that middle quintile there. But you can see that all the people down here, so particularly the top, say 10 or even 20% of people, they will have hit that line up to 10, even 20 years before screening kicks in. So they should have been screened much earlier. And at the other end of the distribution, the women with polygenic risk scores in the top 10% of the distribution, they never reach that line at all. So even though these scores are imperfect, there's quite a lot of information here for defining more effective screening programs. Right, a couple of, so moving on to symptoms and diagnosis. I've just got one study here, and this is in psychosis now. So psychosis often appears in sort of older teenagers, and is a really scary incident when it happens. And the first episode of psychosis gets you into a clinical pathway. That what could happen after that is you could end up with a very mild psychotic disorder, or you could develop schizophrenia and have a lifetime of ill health from that, which is very difficult to control. But at that first episode of psychosis, there are very few predictors of who's going to take which path. And the genetics does seem to be a predictor of that, very modestly. But if you look at the study on the left-hand side, this is standardised mean difference from controls, so the controls are at 0. And these are people that went on to develop schizophrenia or another psychotic disorder. So you can see both of these are elevated above 0, but those that went on to get schizophrenia are elevated still higher. So some evidence of signal there. Similar study at the end here, these were chronic schizophrenics, and there's less signal there. But the one in the middle was completely non-predictive. And the reason for that is because these were patients of African ancestry. And most of our genetic studies have been done in people of European ancestry, white populations, Caucasians. Geneticists use the term European ancestry. And that means that we haven't identified many of the genetic variants that are relevant in other populations around the world. And this is a big barrier at the moment as to how we're actually going to implement genetic medicine, because we've got to do a lot more discovery to make sure that it's available to everyone for equity, not just the simple, easy white populations in Western countries that we've done the studies in. I'm going to skip over that for lack of time. And then one of the things I want to talk about is course of illness. So many disorders, as I just talked about with schizophrenia, it's not clear at diagnosis whether someone's going to recover or have a mild case of the disorder, or whether it's going to be a lifetime of ill health. And that's true for depression, for schizophrenia, for rheumatoid arthritis, for many things that you want to mention. And so and I think this is one of the places where genetics could really be valuable, partly because we're not telling people that they have a very high risk. We're just informing possible treatment and possible outcomes. So getting it wrong is less of a problem. And it can highlight us to what might happen. So one of the worst things that can happen in any psychiatric disorders is a suicide attempt. And suicide is a major problem worldwide, and you have some fantastic research in Australia in trying to prevent and predict suicide attempts. But over 90% of people who attempt suicide have a diagnosis of a psychiatric disorder. And so we looked in the 3 adult disorders we had access to. This was my PhD student, Lee Mullins, who looked at a big international collaboration. And for each case of these, she went back to the original studies and identified information on whether that patient had ever attempted suicide or not. And so this was a within-case study. And then we said, does the polygenic risk score for any of these disorders help predict who goes on to make a suicide attempt? And the polygenic risk scores for schizophrenia and bipolar disorder had no prediction at all. But our scores for depression had some prediction, not only in depression cases themselves, but also in bipolar and schizophrenia cases. And you can see that the Nagle-Cokies R-squared is very small here. The p-values are perhaps convincing, but not enormous. And you can see one of the problems here is that the sample sizes here are really quite small. If you look back at the sample sizes that we had to discover the polygenic risk scores, we had to use 250,000 cases of depression to find our 100 loci that we have that are associated. And that's quite easy to do because lots of people collect information about depression. It's in our population cohorts, it's in clinical cohorts. And so it's quite easy, and because depression is such a common disorder, it's quite easy to accrue those large sample sizes. But when you're looking for incidents that occur within cases of disease, the sample sizes are very much smaller. Now, there's no reason to think that the genetic effects are larger, and so we're really struggling here to find a signal. But we'll search. So this is nowhere near clinical implementation, but I offer it here as a possibility of the sorts of things that we would really like to be able to get towards doing. At the point where our doctors, our electronic health records contains a copy of our genetic information, and anything any consultation that we have can be genetically informed about possible diagnosis or possible treatment. So this is where we are with polygenic risk scores. I hope I've shown you how tempting and how we've started to make progress here. We really need to increase the predictive ability and validate before we can even move towards implementation. And one of the challenges is that we can do our research studies, but to get that accurate for a single individual and know how to interpret it is much more difficult, particularly to make sure that they're valid for everyone. There's going to be a whole lot of communication work that needs to be done here. You saw my reaction when I saw my coronary artery disease risk and was quite worried by that, quite unjustifiably. But there's going to be a lot of information. Also educating doctors. We're used to thinking of genetics as a yes-no, but now we have genetics which is on a normal distribution, and that's a changing concept for many people. So it's an area that's still very much under construction, but this is a nice quote. The outstanding questions is not whether genetic profile scores, they call it, can be adopted in the clinic, but when and how that will happen. And I'm really looking forward to being on that pathway to using it. I wanted to finish with a bit of local interest. This, of course, fits in very nicely with a major new investment here, the Australian Phenome Centre. On the very right-hand box here is our disease risks, which is what we're talking about. Now, the Phenome Centre is doing it through metabolomics and gene expression and diet and such like. I've been talking about this box here, our genes, and going straight from here to here, and that certainly fits in with those boxes there. So I'll finish by coming back to Frank Hansford-Miller. I've given you the quote, "How is this piece of research going to help humanity?" And I hope I've been somewhat convincing of that. And also, I think that we all have the potential to put statistics back in and make a bigger contribution to society. I'll finish by thanking, that's my research group on the back, and the International Depression Consortium on the right hand side there. And I'll just finish with a very big thank you, particularly to Brenton for organising this and hosting me. I love the DNA spiral in Kings Park. It's just wonderful, and I've thoroughly enjoyed my time with you.